Rhys holds a PhD in chemical biology from the University of Southampton, where he worked on cyclic peptide inhibitors. He joined Curve Therapeutics in 2020, where he now is a team leader with a main focus on validating hits through techniques like Spectral Shift or SPR. His expertise on cyclic peptides proves useful, in particular against IDPs. He will now talk to us about his work. Please, Rhys, go ahead.
Thank you for that introduction, Nicola, and welcome everyone to my section of this webinar, which I'll be giving you a little bit of an overview of some of the work we have been carrying out at Curve with a focus on how we've been utilizing our Dianthus. But firstly, a little bit of background. So as many of you will already know, increasingly in recent years, the pharmaceutical industry has been pursuing new target classes, such as protein-protein interactions and transcription factors. And these new targets have brought with them new challenges. The main one being that they've been challenging to approach with the traditional small molecules that we use for things like GPCRs. So in the bottom left corner here, I've just included this little graph of the different drug modalities. We can see we have our small molecules, which are typically, below 500 molecular weight and adhering to the Lipinski-like rules. We then have these beyond rule of 5 compounds, so named because they don't adhere to the Lipinski rules. And these are things like macrocycles and natural products. And so these are a bit bigger, usually between 500 to 1.5 kilodaltons. And these are capable of binding to grooves or clefts in a protein surface. And then in the top right as well, we've got things like biologics. These would be antibodies. And these are anything over the 1.5 kilodaltons. And these are going after the very large surfaces, things like protein-protein interactions. So just at a glance, we can see there's an advantage to these beyond rule of 5 compounds, where we can go after these protein targets that have maybe grooves and clefts, but lacking those pockets required for small molecule approaches. However, conducting drug discovery campaigns in the beyond rule of 5 chemical space presents a significant drug design and development challenge. Curve's approach to this challenge is the development of a novel DNA-encoded screening platform. So now I just want to introduce you a little bit to Curve Therapeutics, tell you about what we do, and explain what a microcycle actually is. So Curve is a spin-out from the Tavasoli lab at the University of Southampton, and was founded a little over 4 years ago, and is built on the development of a novel DNA-encoded screening platform, capable of screening cyclic peptide libraries inside of mammalian cells. So a typical library consists of about 300 million different cyclic peptide hexamers, and it's this small ring size that's very key to the platform. So we found that microcycles, which is any cyclic peptide of 5 to 7 amino acids, possess a compactness, rigidity, and well-defined pharmacophore that make them more amenable to transitioning to small molecule scaffolds. So typically the library is transduced into your cell line of interest, and this will contain a fluorescence sensor within the cell, which is then used to sort your hit sequences. Once the cells containing hit sequences have been isolated, so we usually do that on a fax machine, we can then use next generation sequencing to determine the active cyclic peptide sequences. And so the aim here is to use the biggest size of the beyond rule of 5 microcycles to identify binding sites that are missed by small molecule approaches, screen them inside the environment of a mammalian cell, and then through some extensive SAR campaigns, rein in the size of that hit molecule to try and reestablish the benefits of traditional therapeutic scaffolds. So that'd be cell permeability and oral bioavailability. Alongside this, we're also in the process of building out a microfluidics platform. So this should complement our cell-based system. And this platform utilizes femtoliter-sized droplets. And these act as individual reaction compartments. And we can actually use these to express both the DNA-encoded library, but also the target of interest using in vitro translation transcription. And this approach would enable us to tackle different challenges, so things such as screening against extracellular targets, but also to start to incorporate some non-natural amino acids if we want. And this will allow us to expand the chemical diversity of the libraries we can screen. Again, fax sorting and NGS can then be used to elucidate our hit sequences, which are then made in our chemistry labs. And it's this stage where the biophysics can get involved in validating these hit cyclic peptides in vitro, and then trying to transition them into those smaller scaffolds. So why do we choose Dianthus as our go-to for initial screening of these hits? Well, the nature of Curve's platform means it's well placed to tackle these challenging targets, That, of course, means we need a downstream workflow that is also capable of dealing with these challenging targets. So in addition, Curve was looking to grow substantially in the coming years, so we knew we had to get an instrument that could keep up with our screening demands, and the Dianthus with its 384-well format met that need. Curve had also had a monolith instrument in the past, and so we had already established a couple of assays that we wanted to transfer over to the Dianthus. So it's a good choice for us as we knew it should be compatible with those. Also, the solution-based nature of the assay made it appealing as we had a particularly challenging target that wasn't very happy being immobilized for SPR. But one of the key deciding factors for going with the Dianthus was the straightforward assay development, which in my personal experience has been a lot quicker to get up and running than some other biophysical methods. And it's actually this assay development that I want to showcase in the remainder of this talk with an overview of the typical development workflow here at Curve. And so this starts out with buffer exploration. This is always a very good first step to find out the buffers that keep your protein happy. And it's especially useful for targets that have little to no available literature. So the Dianthus makes this very straightforward. It has a built-in method, which utilizes this 96-well buffer block, which Nanotemper will sell to you. And it lets you quickly screen a wide range of buffer conditions. So what we see on this left-hand side is actually the live readout. So this is what you would see in the control software. And it actually highlights in real time the conditions that look most promising. So the darker cells are giving us the best signal quality. And then on the right, we can do some sort of post-run analysis. And already here we see the software has highlighted in red some conditions that maybe our protein is not very happy in. If you run this buffer exploration in the presence of a control, or indeed the compound of interest, you can essentially screen the 96 conditions for the buffer that gives you the most robust binding signal. And again, the software has a number of visualizations for displaying the output of that experiment, where we have the individual runs on the left here. So that's all the individual signals. And then also it has these tools such as this pie chart visualization, which you can use if you're writing up reports, where it's put these conditions into 4 different categories. So there's non-binder, inconclusive, autofluorescence, or hits. And so this is just a nice way of quickly identifying the promising conditions that might be worth a bit of a follow-up. Once we've found our suitable buffer that keeps our protein happy, the next step in our workflow is always to run a binding check. So this is a really quick way of checking the performance of the assay, where we have a protein alone reference, which is the 3 green dots, and our protein plus positive control, which are our 3 yellow dots. And then hopefully, if you've got a nice interaction, you'll see a good shift of the ratio and also a decent signal to noise ratio. So we can see here we've got good signal to noise of about 19 and a decent shift on the Y as well. And we find this very useful for performing quality control, especially at the beginning of the day, as it allows you to check that your protein batch is good to go before committing to that whole plate setup. And indeed, it can also be used for protein quality control. So if you want to make sure you have consistency between different protein batches. So once we have our optimal buffer and we've checked our protein batch using a binding check at the beginning of the day, we're ready to let the Dianthus stretch its legs a little bit and move on to some affinity screening. Affinity screening is the main method used here at Curve. The nature of the platform means we already have some confidence that our peptide hits should bind to some degree. And so what we need to be able to do is rank those hits by affinity. So there is an option with Dianthus to screen single doses for more of a yes-no answer. And then obviously you could test 384 compounds at once. But for our case, we need those full dilution series to get the KDs. And so here we have actually 30 different compounds in 12.0 dilution, with our yellow wells being controls. So this is actually a screen grab directly from the control software. So this is what it would look like. And again, you get this live data output. So in the top right, this curve of the data points actually appear in real time. So if you're a bit impatient for data like me, it's very tempting to sit and watch your curves appear. And then also on the right in the middle and bottom, we have a couple of control metrics in our initial fluorescence and our well scans. So now we've run our screen, we can move over to DICER, which is the analysis software. And I really just wanted to showcase here a couple of the features we found helpful at Curve, with the main one being the automatic data interpretation. So in this example screen, we can see the bottom condition has been categorized as a scan anomaly. And so in the software, if you double clicked into this, you can get a sort of blown up view of the data. And so in the middle bottom, we can see that we do indeed have a scan anomaly. So something other than one-to-one binding is going on there. And so we do have correct interpretation. And equally, second from bottom as well, if we double click into that one, we can see the software here is categorized as a weak binder, and even in the details told us that there's insufficient saturation for a plot. And again, yep, the software is bang on the money, and we can see we would need further data points to get that bottom plateau on to get a decent fit. And then of course, if you do have a good binding curve, which hopefully is what you see most of the time, then it will categorize it as a binder and automatically calculate your KDs. So you've heard a lot today about Spectral Shift. So for those of you who've maybe only used Trick before and not Spectral Shift, as is the case for me until about a year ago, I wanted to show you this data. So one of the questions we had at Curve was about whether the new Spectral Shift technology would correlate well with the existing Trick data. And so I've plotted here the Trick KDs obtained on the x-axis and the Spectral Shift obtained KDs on the y. And at a glance, we can already see that they are very well correlated. But notably, one thing we did notice is of the 63 compounds we assessed in this screen, 49 gave us a plottable Trick window, whereas all 63 had a plottable Spectral Shift window. So this is sort of coming back to that robustness of the Spectral Shift, where, at least for this protein, the increased sensitivity of Spectral Shift means we identified hits that we otherwise wouldn't have been able to find. So lastly on the data, I just wanted to showcase the impact that these screens are having here at Curve and how we've been utilizing our Spectral Shift data. And so I've put together this plot where we have the molecular weight on the x-axis and the Spectral Shift determined KDs on the y. And the data points are also color-coded, so red's high molecular weight and green is low. And if we start off on the right-hand side of this plot, we see we sort of have our microcycle beyond rule of 5 compound hits. So we're up to the sort of 800 molecular weight on the high end, but a lot of these are around the 700 molecular weight. And some of them are already decent binders, straight out of our cell screening. However, we're sort of at those high molecular weights and we want to be transitioning down. And so what we can see from this plot is indeed we have now managed to sort of drive towards this left-hand side, down as low as the sort of 300 molecular weight point, whilst retaining, or in some cases even improving, on the affinity of these small molecules for the target now. So it's a good way of showing just how Dianthus has started achieving that goal of utilizing the beyond rule of 5 hits and then transition them back down to smaller scaffolds. So to finish off, I just want to say a quick word about Curve's future plans with Dianthus. So obviously we'll continue to use it as our go-to for screening our hits, including the development of some new screens when we have our new targets. But we're also looking at a couple of different applications, including the use of the biotin dye. So Nanotemper have recently released a new biotin labeling method. We're looking to use this to help with the development of our SPR assays, as a lot of our SPR proteins are already avi-tagged, so they're immobilized via a biotin, and this would just allow for a very direct comparison of the two methods using exactly the same protein construct. And then lastly as well, the use of cell lysates. So if you ask your cell team very nicely to develop a his-tagged version of your protein, we can then use the NTA label to probe compound binding to that construct, which has the benefit of being expressed inside of a cell. So obviously, if you have any PTMs or any components within the cell lysate that are important for binding of your compounds, those should be present in the cell lysate experiment.